welcome to Science Friday.
Here's some for E as well. The one on the top here is one of the ways that you can define E.
What makes a field more likely to have an unbalanced gender distribution?
So this can be like, in the case of this paper, gyrification or brain folds.
A neuroscientist started to look at the brain as
like the
more as an
incredibly negative consequences.
Thank you, Kira. So as Kira mentioned, I'm going to be talking about the reprogramming of lipid metabolism in cancer-associated fibroblasts and how that potentiates the migration or metastasis of colorectal cancer cells, which I'm just going to call CRC cells from now on because that's easier. So let's get into a little bit of why I'm specifically interested in this topic. Growing up, my parents are doctors, my grandparents are doctors. There's been a lot of conversation about cancer and just how it grows and frankly kills people. So it kind of became this big scary thing in my mind where someone just wakes up, goes to the doctor and has stage 4 cancer. And I think I became kind of curious about why that happens. And I think additionally, in kind of popular culture and the way that we understand cancer, it is sort of this big scary thing that we don't fully understand, but that is kind of like a big deal. So that's kind of why I'm specifically interested in this, and kind of the question that we want to address throughout the course of this presentation is how does cancer specifically grow and spread, because that's really determinant of its effects on the human body. So let's talk about the most basic answer to the question of why tumors grow and spread, and it's metastasis. As Kira mentioned, metastasis is where tumor cells spread from, I have a laser pointer, very exciting. An original tumor through the lymph system and the cardiovascular system into another part of the body where it forms a metastatic tumor. So basically tumors will start as a primary tumor and then end up being metastases of that smaller tumor all over the body. And metastasis is a big deal because the majority of cancer-related deaths are caused by metastases. And basically, we understand that metastasis happens, but we don't have a complete understanding of how metastasis works or why tumor cells are able to spread in the way that they are. So an important factor in understanding why tumor cells are able to spread the way that they are is the tumor microenvironment. Again, I'm just going to call it the TME because that's a lot of letters. And the tumor microenvironment is basically a bunch of cells that the tumor recruits in order to support its just growing and spreading. So tumors will kind of bring things in like T cells, macrophages, red blood cells, just to basically do the tumor's bidding. They basically feed the tumor, help it grow. And the cell that we're specifically going to be talking about for this presentation are cancer-associated fibroblasts. And cancer-associated fibroblasts are basically critical for providing the fuel that tumors need to grow and spread. So what are cancer-associated fibroblasts? They're normal fibroblasts that have basically been recruited for pro-tumorigenic purposes. And normal fibroblasts are just cells that build extracellular matrix, excrete proteins. They basically do a lot of different things. But when they come cancer-associated fibroblasts, all the factors that they excrete are kind of used to help support cancer cells. And these factors are especially important in determining basically the metabolism of tumors. And there are a lot of different types of metabolism that can take place. And the two that are most the best documented at this point are glycol and amino acid metabolism, but lipid metabolism is not as well documented. I guess before I move on into the meat of the paper, are there any questions about metastasis, the tumor microenvironment, CAFs? Okay. Also, just feel free to interject at any point, and, or raise your hand, and I can take your question. So the thing that this paper aims to address is determining what basically happens when cancer-associated fibroblasts excrete lipids and whether or not they're taken up by tumors, and what that then does to tumor metastases. So that is kind of the goal of this paper, and something that it gets a little like convoluted because there are a lot of different experiments, but that's something that you should just keep in mind for the overarching goal. So in terms of experimental design for this experiment, it's a little bit different than a lot of experiments that we kind of traditionally think about, which are just you do a thing and it proves like what you're trying to prove. This experiment basically takes a bunch of really small experiments that they do, and then they build up to kind of a correlative hypothesis that they then say that they've proven, basically. So everything that I'm going to be presenting from now on are one of the experiments that they did to kind of build up to their big conclusion about the interaction between cancer-associated fibroblasts and tumor cells. And specifically in regards to the experimental design, what's important to understand is that the researchers are basically taking calf juice. So they like culture calves in like a medium, and then they take the juice that calves excrete. I'm just going to call it juice because that's easier to understand. And then they put it in with tumor cells, and then they see what the tumor cells do depending on the experiment. So you're not actually seeing a lot of like co-culturing. It's just a lot of co-culturing of the juices that come out of calves into CRC cells. And just tell me if that's like a weird metaphor that you don't understand, and I can explain that better. But yeah, that's kind of the way that the experiment is working. So the first thing that they set out to prove in this experiment is that calves promote the migration of CRC cells. And they do this by proving that there's an elevated risk of metastasis with cancer-associated fibroblasts versus normal fibroblasts. So they first examined proliferation, which is like tumor growth, not necessarily tumor movement throughout the body, but just tumor growth. And they found that there was no difference between cancer-associated fibroblasts and normal fibroblasts in terms of CRC proliferation. However, the difference that they did find was that calf-conditioned medium, so calf juice, induced CRC metastasis at a higher rate than normal fibroblast conditioned medium did. So from this experiment, they basically determined that there's something in the calf juice that cancer-associated fibroblasts are excreting that is then taken up by CRC cells that causes metastasis. So the rest of their experiment kind of set out to find what this factor was. And they eventually settled on lipid metabolism. So they found that cancer-associated fibroblasts secrete more lipids than normal fibroblasts. So if you look at this graph, all of these things on the bottom are different lipids. On the side, this is their relative abundance, and the black graphs are cancer-associated fibroblasts conditioned medium. That's what they're calling the juice, and normal fibroblast conditioned medium. And you can see that there's an elevated level of fatty acids fatty acids in lipids for pretty much all of the ones that they tested. So what this means is that there's a real potential for the correlation of, of cancer-associated or tumor cells taking up lipids and then inducing metastasis, but they haven't proved that yet. So they kind of try to get a little bit more specific in proving that this happens in the body. So since they've proved this correlation outside of a like real tumor cell environment, they're basically kind of simulating the what happens in tumor cells. They basically set out to determine whether or not this interaction could happen in actual tumor tissue. So they do a comparison of normal tissue, which is this part here, and tumor tissue. It's a little hard to see on the screen, but they basically stain here for cancer-associated fibroblasts. That's alpha SMA, which is like a marker of calves. And they saw that in the CRC tissue, there's a greater abundance of calves. And additionally, there's also a greater abundance of FASN, which is fatty acid synthase, the thing that makes fatty acids, which is basically what they're testing here. So from this graph, they basically found that the interaction that they characterized here happens or is relevant to CRC tissue. So the next thing that they did is they had to prove that it's specifically lipid metabolites that are taken up by CRC cells. Because at this point, all that they really have is a hypothesis that lipids are taken up by CRC cells, and that's what induces metastasis. So the first thing that they have to do to prove this is to prove the first part, that lipid metabolites are indeed taken up by CRC cells. And they do this by doing a comparison of normal calf conditioned medium, which is just calf juice, the stuff that it excretes, and then a comparison of conditioned medium, where the conditioned medium has been put in with CRC cells. So what they're basically testing is the difference in the conditioned medium before and after CRC cells, meaning that they're examining what the CRC cell is taking out of the conditioned medium. And since this kind of ratio is negative here, this means that CRC cells are in fact taking lipids, specifically fatty acids, out of calf conditioned medium. So that's proof for like how lipids are taken up by CRC cells. And finally, what they did is they proved that preventing lipid uptake prevents further metastasis. So that's kind of like fully finalizing their conclusion here. And to be honest, like this graph was super unclear in the paper, like what 1, 2, and 3 are. The best guess that I had was that they're different trials, and they don't specify the difference between these two red and white here. But basically what this graph is telling you is that the black is normal cells that have normal cancer-associated fibroblasts that have been conditioned, normal fibroblast conditioned medium that has been cold cultured with CRC cells that they then examine the metastases of. So this means the black, you're seeing that there's a higher rate of migration in tumor cells that have fatty acid synthase. And then in cells where fatty acid synthase, the thing that literally makes fatty acids, is knocked out, there's less metastasis. So this means that a deprivation of fatty acids in the tumor microenvironment and then being taken up by CRC cells leads to a decrease in metastasis. So that's kind of this is kind of their final conclusion. And this kind of leads us to an updated understanding of how we kind of characterize the interaction between cancer-associated fibroblasts and CRC cells. So kind of the kind of big conclusion from the study is that FASN synthesizes lipids, which is then taken up by CRC cells and leads to an enhanced migration ability. The last experiment that we just talked about is where they knock out FASN in calves, and then there's a decrease in the lipid. Basically, there's a decrease in lipids that's taken up by CRC cells, and that leads to a reduced migration ability. So that's kind of the study is contributing to our understanding and the way that we can understand how tumors basically work and spread and eventually can become lethal. So why this is important is, again, understanding the mechanisms by which tumors spread is essential for kind of new treatments and providing new ways that we can address tumors. And even still, we see that more research and more demystification is necessary, because we've basically established sort of a strong correlation in the study. They do find some strong evidence for a lack of fatty acids leading to a decreased potential for metastasis, but we really don't understand the mechanism by which that happens. So we don't actually have a treatment yet, because we don't know exactly what we're doing by targeting FASN, because we don't have the full picture. So this is why kind of demystification and kind of the curiosity that we talked about at the beginning of this presentation is important for like real world science and understanding treatments. So yeah, that is all I had. Thank you.